Welcome to Decoding Healthcare Research. I'm your host, Efrain Riveros, Dr. E.F. Rain. I am an associate professor of anesthesiology at the Medical College of Georgia in the United States. And today we are going to deal with a very interesting topic that has to do with brown adipose tissue and exercise. So we have the honor of having with us Dr. Guillermo Sanchez-Delgado. He's a researcher from the University of Sherbrooke, Canada, and also University of Granada in Spain. So welcome, Dr. Sanchez-Delgado.
Thank you very much, Dr. Riveros. It's a pleasure to be here.
Thank you so much. So we are going to be discussing a paper that was published in Current Diabetes Report in 2020. The title of the paper is Endocrine Mechanisms Connecting Exercise to Brown Adipose Tissue Metabolism, a Human Perspective. So this is a topic that is becoming of interest, to many people in the context of exercise, also health and disease. So the brown adipose tissue, and that's going to be the first question. What is the difference between white adipose tissue and brown adipose tissue, and why is this becoming a new trend?
Yeah, I think that's an excellent question. So brown adipose tissue, as the very name says, is pretty much an adipose tissue. So we can say it's rather similar to the white adipose tissue we have all over the body, right? The main difference, and that's probably why we call it brown, is that the brown adipose tissue is really vascularized. It's really active from a metabolic point of view. And probably the hallmark, like the key difference, is that brown adipose tissue has a lot of mitochondria, or brown adipocytes have a lot of mitochondria. And these mitochondria express what we call the uncoupling protein 1. This is a, let's say, very particular protein, which basically uncouples ATP production from the oxidative metabolism, for the electron transport change. So basically, in a nutshell, brown adipose tissue is an adipose tissue that, thanks to these mechanisms that I mentioned, is able to generate heat. Basically, it consumes a lot of energy, and as a result of that, we create or generate heat to maintain body temperature. So just to say that the huge difference, I would say, besides anatomy and all this stuff, is the function. The function of white adipose tissue is probably mainly storing energy and releasing energy when we need it. For brown fat, for brown adipose tissue, the main function is to generate heat to maintain body temperature.
And it's interesting, when I think back when I was in medical school, brown adipose tissue was mentioned only in the context of neonatal life as a source of heat and regulation of temperature in neonates. And that was something that we knew back then. But lately, there have been multiple studies demonstrating that there can be a transition between white and brown adipose tissue in other contexts. And you mentioned that it has to do with the mitochondrial function that is increased in brown adipose tissue. So let's talk a little bit about that transition between the two types of adipose tissue.
Yeah.
How does that happen? Can white fat become brown fat?
That's an excellent question. And actually, going back to the start of your question, why 20 years ago, we were not talking about brown fat in humans. Now we are doing that. The reason is, and I'm pretty sure we will touch on that later in the conversation, when they were starting to use the PET-CT, that's an imaging technique mainly used in oncological settings, they started to see something that is symmetrical, something that looks like adipose tissue, but it has a high metabolic activity. Back in the time, and talking about the beginning of this century, we believe that, as you said, adults don't have brown adipose tissue. But investigating that, we actually discovered, and that was 27, 29, 14, 15 years ago, that we discovered that adult humans have brown adipose tissue. So that's the reason why we are now talking about it, and we were not talking about it before. Now, about your question, I think that's also really interesting. Can we transform white fat or white adipose tissue into brown adipose tissue? It's a yes and no answer. So first, I think we'll need to say we were talking so far about white adipose tissue, brown adipose tissue as two different compartments or two different type of cells. Let's call it like that: white adipose tissue, white adipocytes, brown adipocytes. But we should introduce something that is kind of in the middle. It's what we call beige adipocytes. Back in the time, they were also called bright, from brown in white adipocytes. So now, to your question, what we do know is that there are processes by which white adipocytes can be, let's say, transformed in those that we call beige adipocytes. So those beige adipocytes are brown-like. They are not brown adipocytes, and we will discuss that in a second, why they're different, but they are kind of like the same. Let's call it like that. They're very similar in terms of anatomy, function. They do express the UCP1 protein, and we can, as I said before, we can transform white cells or white adipose tissue, white adipocytes into beige adipocytes. Therefore, we do have this process that we call browning. So basically, when you submit a participant or an animal to certain stimuli, for example, cold exposure, you will see that browning. So part of what was before white adipose tissue now looks brown, now looks beige adipose tissue. Now, another big question is whether this is transdifferentiation of mature cells or whether we are actually seeing differentiation of new cells, so basically proliferation of new cells. Probably the two phenomena takes place, but for sure, at least from a, let's say, histological point of view, there is a browning. So browning happened, browning exists, and this is the transformation of a white-like adipose tissue to a more brown-like adipose tissue. That to say, however, as I said before, brown adipose tissue or brown adipocytes and beige adipocytes, even if they are very similar, they're not the same. And actually, ontogenically speaking, they're different. So that's why I'm saying that you cannot get a white cell and make it brown, but you can get a white cell and make it beige, which is functionally pretty much the same. But no, that's why I said at the beginning, it's yes and no. It's so we can do browning.
Yeah, and that's actually a great way to present it. That by increasing the metabolic capacity of the adipocyte, you can turn it into a beige cell, and because this has been associated with thermogenesis, there are a lot of studies, evaluating the effect of thermal stress, specifically say cold plunges that are very popular nowadays, and how you can induce that process of turning those white cells into beige adipocytes. So my question is, people tend to think that if you do that, then you probably lose weight or something. I would like to know what is the physiological benefit of having that browning of the white adipose tissue.
Yeah, this is the $1 million question. And we still don't have a very good answer, I will say, even though you will read in the press and a lot of things about it. I think we still don't have an answer. What we do know is that we have consistently, many groups around the world have consistently observed that there is a correlation, an association between the presence of let's call it brown fat, let's call it brown adipose tissue, because we actually are not sure whether what we see in human is brown or beige, probably beige, but anyhow, let's call it, for simplicity, let's call it brown. So what we do see is an association between the brown adipose tissue presence that we can detect and a healthier metabolic profile. So the more brown fat you have, the healthier you look. That's an association, but from an association, we cannot infer causality. We cannot be sure whether actually brown fat is the reason. Now, there are reasons to believe that brown fat might be a contributor to that, let's say, more healthier metabolic profile, right? And I will say, I think there are mainly three reasons. One, brown adipose tissue or beige adipose tissue, it's a really thermogenic tissue. So it consumes a lot of energy, like really a lot. So originally we thought that that could basically increase your energy expenditure, either resting energy expenditure, your diet-induced thermogenesis, and therefore probably preventing obesity. That was one of the reasons that we believe, probably at least at the beginning, that could explain that association. The second one is because the capacity of, or let's say the uptake of glucose, fatty acids, of nutrients by those brown fat cells when they are activated is so high. We also believe that it could be that when we are exposed to let's say a high nutrient availability, like for example after a meal, you have high glucose, high fatty acid, not fatty acid, let's call about glucose. So then beige or brown fat could be taking that like a sink and therefore preventing hyperglycemia or like hyperlipidemia in other conditions. So kind of this second, let's say second hope or second reason to believe that brown fat is actually protective is this sink, kind of sink effect, let's call it like that.
Mm-hmm.
And now the third, and that's, I would say this is probably the more unknown so far, is the endocrine function of this tissue. So we now are pretty sure that brown adipocytes or brown fat secretes molecules. It could be proteins, it can be lipokines, it can be microRNAs. It is an endocrine active tissue. Some of the molecules that brown adipose tissue secretes to the bloodstream are also related to let's say a healthier profile. Some of them are well characterized, and we know they have an effect. For example, FGF21, that would be probably the most canonical one. It improves, for example, insulin sensitivity in peripheral tissues. So that could be, again, another reason why brown fat might be healthier or might be protective or might be beneficial for your health. But notice that I'm saying it seems, it could be, because actually we still don't have any proof, at least in humans, that brown fat actually improves metabolic health. We don't know.
And that is actually the best way to present it, because if the brown adipose tissue is related or associated with a healthier status, I assume that people started thinking that because exercise moves in the same direction, there could be an association between exercise and the browning of the white adipose tissue. So where is exactly that connection? That is what our discussion is leading us to. So what is the connection between exercise and brown adipose tissue?
Yeah, that's a complex one. And I will tell you the story. So 29, now the world, we all learn that brown fat is present in humans. We are translational scientists, so we do human research, actually. So we get interested on that. I mean, I would say everybody got interested on that back in the time, right? And our background is sports science. So we started thinking about exercise. And the reason why is because there were quite a few studies in preclinical models, so mouse, rats, that actually show that exercise produce browning. So remember this process we were talking about before. Browning is that process by which we can transform white fat into brown-like or beige fat. So this browning was clearly demonstrated in response to exercise in preclinical studies in mice, for example. Now we should go back to that and discuss whether this is actually the case or not, because there is a intricate, complicated picture. But back in the time, I'm talking about the beginning of the last decade or the past decade, that was the picture. So preclinical studies were telling us exercise seems to induce browning. Then we all were wondering why or how, and that's a mechanism you're probably thinking on, is those endocrine signals that are up or down regulated during exercise that are part of the process of browning, so they can induce browning. So back in the time, for example, there was a really, really famous paper. I think it was published in Nature, although I don't remember. That was from the group of Bruce Spiegelman in Harvard. And basically what they showed to the world is that this new molecule with a fancy name, irisin, that was a protein that was secreted by skeletal muscle during exercise. Irisin induced this browning process we were talking about. Now, but I mean, 10 years later, we know that the picture is not that simple. When you look at the most modern preclinical studies, we're learning that that clear effect that I mentioned before of exercise on browning is not that clear, actually. We only see, or apparently, we only see that effect when mice are exposed to room temperature. Now, a mouse, when you put a mouse in room temperature, it's far from thermal neutrality. So basically, it will let's say, it will be equivalent for us to be on a cool room. So now, when mice do exercise in that conditions, browning is produced. That's pretty clear. Now, however, some later studies did that question, and basically they tried, will we see the browning effect of exercise if mice are placed at thermal neutrality? And apparently not. So it seems to be an interaction between exercise and cold exposure. But this is what we know now, not what we knew when we started working in humans, because we started doing that question. Is it also happening in humans? Does exercise activate or recruit brown adipose tissue? And basically, we devoted like 4 or 5 years of our career to answer that question.
Yeah, and it's very clear that from those models in mice, we started exploring what's actually happening in the human body, and I think the value that I find in your paper is that you try to answer that question: what is in reality the connection between exercise and browning of white adipose tissue? And you actually put together a very nice review of the different substances, because you mentioned that the endocrine and also paracrine network that is involving the adipose tissue, the muscle, and even other tissues has to be explored from different angles. And that's exactly what you did in your paper. So I want you to go over the different molecules that have been found to be part of this intricate network of metabolic mechanisms that lead to browning of the white adipose tissue, being the first one of them norepinephrine. What can you tell us about that?
Yeah, actually, I think you picked the right one to be the first. And norepinephrine, it's the main physiological conditions, it's the main activator of brown adipocytes or brown adipose tissue. And we know that in response to exercise, especially if the exercise has, let's say, some level of intensity, we do secrete norepinephrine. So that actually was one of the first that make us believe that exercise could actually recruit brown adipose tissue. That norepinephrine, we know that it recruits and activates brown adipose tissue, and it's also secreted during exercise. Now, like for example, we could have an example. I think this is one of the most beautiful examples, or let's say more promising situations for brown fat to have a role on metabolic health, right? Because we didn't mention that before, but the volume or the quantity of brown fat that we see in the human body is rather low, at least in, let's say, normal Western living conditions. Now, why I was saying that, sorry. Yeah, norepinephrine. If you have a patient with a pheochromocytoma, which is a kind of tumor that secretes a lot of norepinephrine, wherever the pheochromocytoma is placed, you see a lot of brown fat. So that is basically the proof of concept that norepinephrine can induce browning, actually can induce a lot of browning. And again, norepinephrine is secreted by exercise. So that's why we started thinking that norepinephrine could be one of those. But then, as I mentioned, there was not only norepinephrine, there was another, let's say, exerkine. They use that word to refer to any molecule which is secreted in response to exercise. So exerkine, another exerkine that really at the very beginning of that was already on the conversation was the atrial natriuretic peptides. So this is a protein that is secreted by the heart. And again, it's secreted by the heart when you increase your heart work, your heart rate, your basically heart functioning. And again, this, let's call it AMP, atrial natriuretic peptide, can again induce browning. So again, we we found a second reason or a second molecule that could actually induce browning in response to exercise. And as I mentioned, we were talking about the sympathetic or the nervous system as the source of norepinephrine, now the heart as the source of adrenergic peptide. And this is when the paper I mentioned before came, the paper of the irisin paper. So this paper that put the muscle in the center of the picture and told us that there is this new protein, irisin, that is secreted by skeletal muscle and could go to your adipose tissue and do this browning process. Now later in the, you go ahead, I'm talking too much.
No, you're actually good at explaining everything in detail. I just want to go back a little bit to norepinephrine because you mentioned the example of pheochromocytoma. So the norepinephrine that is able to induce the browning is circulating norepinephrine or catecholamines, or is like a synaptic at the terminal of the synapses, the one that is secreted?
That is an excellent question, because indeed the activation of brown adipose tissue is not through circulating norepinephrine. So you will see brown fat activated without any increase in circulating norepinephrine. It's synaptic norepinephrine. Now, if you use norepinephrine like in vitro, you will induce browning, that's for sure. But I think your question is really good. Like, will circulating norepinephrine has an effect on brown fat or browning? At least myself, I don't know. But that was kind of one of the, let's say, first problems we found when we were thinking on this kind of hypothesis of exercise activating brown adipose tissue. It's not the same, and the norepinephrine that circulates in the bloodstream than the concentration that the let's say the adipose cell sees in the synapses, right, of the determination with the nerves. Yeah.
My second question in that regard that has to do more with the molecular mechanisms and what happens in the brown or in the beige adipose tissue probably. What happens when that norepinephrine activates the receptor in that tissue? What actually is the sequence of events that that explain the actual increase in thermogenesis in that cell?
Yeah, I think this is a really timely question because, and I think this is a good example of the, let's say, the novelty of this field. So, as I mentioned at the very beginning, there are more unknowns than knowns, and I think this is a good example. So, the canonical pathway, the canonical, let's say, the classical way, I will have explained that 5 years ago, is that norepinephrine will bind to the beta-3 adrenergic receptors in the surface of the adipocyte, and then by a kind of a long, let's say, pathway, it will activate lipolysis. And this activation of lipolysis, this increase in the intracellular fatty acid concentration, will in turn activate UCP1, so activate brown fat thermogenesis, and also promote the transcription of some genes that will, let's say, favor the proliferation of brown fat, or in other words, favor the hypertrophy or hyperplasia of the tissue. But that's basically the connection. So norepinephrine will bind beta-3 adrenergic receptor, activate lipolysis, lipolysis will go and activate UCP1. Now, I was saying at the beginning, this is a good example of why we're learning so much, because we're working on brown fat. I mean, and when I say we, I mean all the scientific community. We're working in brown fat only for 12, 14 years in humans. Just a couple of years ago, we learned that this picture I just described, which is rather accurate in murine models, in a mouse is exactly as I said. In humans, it's not exactly like that, because actually, when you treat, when you use beta-3 adrenergic receptor agonist, you don't see the effect I was kind of explaining. Some of my colleagues here in the University of Sherbrooke, they conducted a study a couple of years ago, I believe, by combining some human studies, but also some in vitro or ex vivo experiments. They actually observed that apparently the most relevant receptor for the action of norepinephrine in human brown fat is not this beta-3 that I mentioned before. It's probably the beta-2. That's another adrenergic receptor. And of course, that changed the whole picture, right? Because like for some years, we have been, I mean, so many people have been looking for beta-3 adrenergic receptors as a potential therapeutic or pharmaceutical to target brown fat. Now that we learn that apparently the expression of the beta-3 is not that much and it's beta-2 the one mediating the effect, of course, we need to switch again the focus and look otherwise, right? So again, as I mentioned, this is a good example of how often we find in the field of human brown fat something that changed the way we were thinking before.
As I went over the paper, and we are going to discuss the different molecules involved in this process, in some places I found the expression of the phenotype of M2 macrophages. So is inflammation involved somehow in the transition between the different types of adipose tissue and the interaction with the muscle?
That's a good question, and I'm not sure I know the answer, but we do know that the macrophages are involved somehow in the browning process because they do secrete some molecules that will create, let's say, the microenvironment for browning to happen. So, for example, let me mention another molecule. I think this is not related to exercise, not that we know at least. It's called CXCL14. This is a molecule that will be secreted by macrophages in the adipose tissue, and it will secrete, it will induce browning. So definitely the immune system and the immune cell infiltration in the tissue plays a role in browning. Now, whether that's relevant in the context of exercise, I don't know. Because, I mean, to start with, I'm not even sure whether exercise induces browning. As I said at the beginning, we believe that back in the time. Now, when we're looking at the evidence, we're not so sure because, sure, there are a lot of molecules, a lot of, let's say, endocrine connections that could support that notion that exercise induces browning. But on the other hand, if we think from a physiological point of view, as I mentioned at the beginning, brown fat is a thermogenic tissue. Its main function is to generate heat. And now let's think about exercise. When you have muscle contraction, you also generate a lot of heat. So why will a thermogenic stimuli like exercise, like a situation in which you actually need to dissipate heat to maintain body temperature, why will it increase another thermogenic process, browning? So, I mean, this is a reason to say that kind of a word of caution, we still don't know whether exercise induces browning. So even if we can speculate about molecules up and down, those could be potential mechanisms. I'm not sure those are actual mechanisms.
Yeah, and that is like the common theme, right, in your paper, that even though there is evidence that during exercise some of these molecules are increased, and in murine models it's been demonstrated that these same molecules are involved in the process of browning, is really difficult to establish that like a causal connection, and that is something that I want you to explain in more detail after we go over the different molecules. You already talked about norepinephrine, natriuretic peptides, and irisin, but there are some other molecules. So in order of importance, which would come next?
That I don't know. Yeah, I don't know. I mean, let's pick one. I would say, for example, FGF21. FGF21, okay. I mentioned that before. I don't know whether this is the next one in importance. I don't know. But I will argue this is a rather important one. And the reason why it's FGF21, it's a really complex, let's say, or the endocrine regulation of FGF21 is rather complex. FGF21 is mainly secreted by the liver, but muscle, under some circumstances, can also secrete FGF21. Now, FGF21, that's pretty clear, induces browning. And to make it an even more beautiful picture, FGF21, as I said before, it's also secreted by brown fat. So here we have a molecule that induces browning, and it's secreted. Normally, most of that is secreted in the liver, but then you also have the muscle secreting that, and you have even the brown fat secreting that. Now, we know that because, well, actually, I mean, at least myself, I don't know whether this is the liver or the muscle, but we do know that in exercise, we see an increase in FGF21. So again, that could be a mechanism or another molecule in this kind of endocrine cocktail that could mediate this exercise effect.
So you think that when the adipose tissue starts producing these molecules like FGF21, there is an effect on the muscle. So it's not just that the exercise can affect the brown adipose tissue, but the other way around as well.
I think that will depend on the situation, but yes, during exercise, I believe that most of what we see in blood is either coming from the liver or coming from the muscle. So in that particular situation, during exercise, I think we should think about brown fat as the receiver, let's call it like that, of FGF21. And that will be the muscle or the liver producing FGF21. Now, in different situations, that might be a different story. Like, for example, we have found that, so we did a kind of a similar study or a similar, let's say, investigation, but the other way around. Now we were thinking on brown fat as the secretion. So now we were interested on batokines. Those are these endocrine signals that I mentioned before that are secreted from brown fat. And our cells, as well as other groups, we do see an effect of cold exposure on FGF21. And this increase in FGF21 during cold exposure, you could say, yeah, it might be muscle again, because when we cold expose someone, we sever. So severing, this is also muscle contraction. That could be the reason why we see FGF21. And it might be, I mean, in part, but we and others, we have found that there is a correlation again between the amount or the increase in FGF21 in circulating in blood and your brown fat volume. So the more brown fat you have, the higher the increase in FGF21 in response to cold. That is suggesting, although we still don't know, but that in humans, upon cold exposure, brown fat is actually producing FGF21, and we should assume this FGF21, of course, is acting elsewhere, not only in brown fat.
Mm-hmm.
So I would say, to your point, depending on the situation. But during exercise, I don't have the data to know whether brown fat is secreting or not FGF21, but I would say most of that is coming from the liver and the muscle. That we know.
And it's fascinating, and so is the effect of IL-6.
Yes.
Understanding that that was probably the first myokine that was identified, and what is the effect of IL-6 on adipose tissue?
That's a good question, and I don't know the full answer. What I do know is that it also induces browning. So for the topic we're discussing here, it's another example of another molecule that you put it in a petri dish, white cells, you put IL-6, and you will see a beige adipocyte, or let's say a brown-like phenotype. And we also know that IL-6 is secreted by muscle during exercise, and actually it's secreted in huge increases. Like it can increase your level on the exercise, can increase IL-6 level by several order of magnitude. So the effect is massive. And again, when it comes to adipose tissue, IL-6 induces browning. So we also put that IL-6 in the list of molecules that could be mediating this effect.
Yeah, and there are several others, and the review is excellent, by the way. I want to focus on one that caught my attention, and is the brain-derived neurotrophic factor. And I bring this one up because I have been reading about the effect of this one on mitochondrial function in the context of Alzheimer's disease. So it basically, I'm very interested in that one in particular. So what is the relationship between this molecule and browning?
Yeah, again, it's pretty much the same story that we were talking before about IL-6 or irisin. Those are molecules that, at least ex vivo, like in vitro, it will induce by itself, it will induce browning. Why and what's for, I don't know, right? And now when we back to exercise, BDNF is secreted during exercise. I don't know whether, I mean, the relative proportion of BDNF, circulating BDNF, that is secreted by the nervous system and which is secreted by muscle, but that we do see increases in that molecule in response to exercise. That's a fact. And again, in the adipose tissue, it induced browning. So again, another one to the list. We believe, or back in the time, we believe that BDNF might also be, besides these more, let's say, canonical effects, or I mean, this is brain-derived neurotrophic factors. So we were talking about neurogenesis and everything. That kind of thing is what came to mind when we were talking about that molecule. But when we were diving in the literature, we started thinking, yeah, maybe besides that effect, it's also playing a role on browning or on adipose tissue browning. Yeah.
Excellent, excellent. And the list actually goes on, and you show the evidence associating each one of these molecules with the browning process. But one that is also important to me now is the ketone bodies, beta hydroxybutyrate. And this is important in the context of exercise and in other contexts as well, like with fasting, ketogenic diets. So what do you have to say about brown tissue and brown adipose tissue and this molecule?
Yeah, the body of literature that I know, at least that we found when we were doing the review, is not that, let's say, big for ketone bodies that it was for another molecules. But there were studies showing that the beta hydroxybutyrate will be able to induce browning. Also in more physiological contexts, I can remember at least a couple of studies that these show that intermittent fasting or that kind of ketogenic diet, so basically not let's call it supraphysiological effects or supraphysiological doses or whatever, but just physiologically or in physiological context, they observe browning again in response to intermittent fasting, for example. And they were talking about these ketone bodies as the mechanism. We don't know whether this is actually the only mechanism playing there, but at least again, ex vivo, in vitro, the ketone body beta hydroxybutyrate can increase this browning process or can induce this browning process.
Excellent. So, and I want to start closing by asking you probably the most important question. After putting together all these molecules that are interacting either in an endocrine or paracrine fashion, what do you think can be said about the effect of exercise in humans on the browning process? So what is safe to say?
Yeah, I will say a couple of things. First, it's just some thinking. It's an idea, not any, let's say, evidence-based conclusion. And it's that we were talking about a lot of molecules that are secreted by exercise or during exercise to the bloodstream, and that we know that in isolated conditions can induce browning. But if we look at the bigger picture, during exercise, we also should be thinking not only about the concentration of whatever it is in the blood, but also blood flow on the tissue we're thinking of. So, and remember what I said before, the first preclinical data was saying, yes, exercise induced browning. But I said to you, those studies were conducted at room temperature. When the mouse is exposed to cold, and that's pretty clear, when the animal is exposed to cold or is cold stimulated, blood flow in brown fat will be upregulated. So basically here you're creating a condition that exercise is producing endocrine signals that can induce browning, and you are opening the door of brown fat. You are increasing blood flow over there. So you're bringing those molecules, let's say in huge quantities, to your brown fat, right? Now, the studies also conducted in mice that do exercise or investigate exercise at thermoneutrality, they don't see an effect. And I think that's probably telling us something about it. If you don't increase blood flow, if you don't open the door of brown fat, it doesn't really matter that the concentration of these molecules is a little bit higher because of exercise. So at least you don't see an effect. That, as I said, it's only speculation. I don't know whether this is the blood flow or not. I think it makes sense to believe that blood flow plays a really important role here. But as I mentioned, this is more some sort of thinking than evidence. But now to your question, what can we say about exercise and brown fat in humans? I think for that, the kind of the best piece of available evidence is a large randomized controlled trial we conducted. Actually, this review we have been discussing about was performed in the context of that large study, large randomized controlled trial. We call it the Activate, but with B, like a brown fat. So basically here we conducted a study. We randomly allocated young adults to 3 different groups. One is a sedentary control group, so standard of care. Second will be people allocated to combine endurance and resistance exercise at moderate intensity. And another group was allocated to high or medium to high intensity training. So basically, that was almost 150 people, so approximately 50 people per group. And the training program was 6 months long. So basically, we investigated whether this exercise intervention that again combines endurance and resistance exercise modifies brown fat in humans. Our hypothesis was that we will see an increase in brown fat volume, probably an increase in brown fat activity. The true, a few years later is that we didn't. Actually, the results of that study that we published, I think, probably a couple of years ago, I believe, in Nature Communication, is that we don't see any effect of brown fat, of exercise on human brown fat. Now, it's because there is no effect to find. That could be one reason. Because when it comes to brown fat, we haven't talked about it, but I think that's very important to keep in mind. When we talk about brown fat, mouse and humans are extremely different. First, because of the body size and so many other reasons we could be discussing, but to keep it simple, they're really different. So even if something happens in mice, we cannot infer that that will happen in humans when it comes to brown fat. So the first interpretation could be there is no effect of exercise on brown fat. Second reason might be something similar to what we were discussing before in the preclinical setting. It could be that exercise could increase browning only if it's performed up during cold conditions. So if you stimulate brown fat at the same time. We haven't done the study to know that, so we don't know whether the results will be different. But exercise performed in, let's say, thermoneutrality, it didn't move the needle when it comes to brown fat. And the third point I think to keep in mind when we are interpreting these results is the way we measure brown fat in humans. I mentioned at the very beginning this PET-CT, this imaging modality, which is kind of the way everybody assess brown fat in the human body. It's a fancy tool. We are all happy about it. We know that brown fat exists because of that. But now that we know more about brown fat biology, we know that this technique has severe limitations to assess brown fat. First of all, because most of the PET-CTs that we all conduct are using an analog of glucose, what we call fluorodeoxyglucose. So basically what you just see is how the tissue takes up glucose. Glucose is a really minor contributor to the brown fat thermogenesis and to the brown fat machinery. So basically you're looking at a big picture only by looking at a corner of the picture. So this is a pretty big problem for assessing brown fat. So this is to say that could be another reason why we didn't observe any effect of exercise on brown fat. I mean, it could be because the tools we have for measuring brown fat, either the quantity or the activity, are still really problematic. And that's why I believe this is part of the reason why in the brown fat field, in human, we still have so many questions because we still don't have very good methods to measure brown fat.
Yeah, and there are definitely gaps to be filled in this field, and I appreciate all the work that you and your group are doing to shed some light about such an important process that is important not only for exercise but in other dimensions related to health. And this has been a very comprehensive review, and I want to, before we close, I want to thank you Dr. Sanchez-Delgado, for taking the time to share your wisdom with us and to shed light about this fascinating field of research. Thank you so much.
Has been my pleasure. Thank you very much, and good luck with this amazing initiative of the podcast. Thank you.
Excellent. Thank you. Thank you so much. And for our audience, you will find the link to the paper in the description of the video. And don't forget to give us your feedback. See you in the next episode.